as my little grandson says, everything I do is an adventure. Getting here was an adventure, I can tell you. So what I'm going to talk to you today about is really a big picture phenomenon about why we're doing what we're doing and why you guys are suddenly interested in some of this microbiology. So these are the folks that do the work. I sit in my office. These are the three scientists and the students that we have doing the work. Dr. Saldias is here. We have Dr. Shmailas Ali and Dr. Sabita Kanasamdi, who is doing some of the work that we'll be reporting on today. But I'm going to talk to you about why has soil biology become a hot topic of interest in science. Well, heck, I've been in this business for 4 years, and until now, nobody gave a hoot, and I've been working on this for a long time. And then we talk a lot about microbes, but do we have any evidence that they really function in a major way in large agricultural systems? And so that I want to present to you that, yes, there is some really good evidence that pursuing this area has some validity. Okay, so let's go back a little bit about how we got to this situation of where we are in agriculture. The second green revolution, the first green revolution was actually built on chicken poop. Or bird poop at least, that was used as a guano that was collected from the world as a source of nitrogen. And the second green revolution came out of the First World War. And there's a gentleman named Haber Bosch, two people, who developed a way of converting, producing ammonium nitrate specifically, and this was nothing for agriculture. It was meant for this. We had a war. And so we had all these factories producing ammonium nitrate to blow each other up, a very important function in society. And then after the war, all these factories are there producing ammonium nitrate. Now we're not going to blow up each other for a while. What are we going to do with this stuff? Well, they started bagging it and putting it onto farms. And out of this came some major benefits to agriculture producer. And we had our first winner of a Nobel Prize for food production, Norman Borlaug. And what he did was he incorporated a gene that stunts the size of plants so that they don't get so high that they blow over. And this allowed wheat production to increase by a huge amount. By the way, no other Nobel Prizes have ever been awarded for producing food. So forget about it. You're not going to get one. Okay, so when we look at nature, what we find is that if you look at the average net primary productivity of an ecosystem, you will find that estuaries, swamps, marshes, tropical forests are significantly produce more biomass than agriculture, which is right in the middle. And then we've got some extreme deserts that are not very productive. So one day my son asked me, hey Dad, how come these places produce, who fertilizes these things? And so there's a question. We can get very high productivity without our inputs because nature has already created a method of producing high yields. Okay, so except for agriculture, we have productivity zones that are based on natural cycles. So, what have we reached? And this is the crux of what everybody here is going to be talking about. It's called nutrient use efficiency. And what we see here is that since 1960 to now, we have lost about 47% of the nutrient use efficiency of your crops. I don't know how many people have farmed here since 1960, but in any event, this has been a declining trend. So what this has led to is, in essence, what you're seeing here is the increased use of synthetic nitrogen fertilizer has reduced the competency of your crops to actually utilize those nitrogen sources and fertilizers. And 43% is a significant loss. So the more we put on, the less benefit we get. And that's just driven up cost without necessarily the return on investments. Okay, so here's the crux of what has happened with all this synthetic nitrogen use. Because you can't just throw these nitrogen sources into soil and not pay a consequence. And what we have reached is we have destroyed a third of the Earth's farmland in the last 40 years. And as a consequence of this is the nutrients use energy that we have lost. And soil is being destroyed 100 times faster than it can form. And to avert this disaster, farmers must adopt sustainable agriculture and based on ecological principles. And this is what your company is doing. And this is what many companies are starting to realize. And many growers are starting to realize also. And lo and behold, for whatever reason, Trump was not doing anything this day, and the New York Times printed an article called, "We Need a New Green Revolution." I don't know what he was doing, maybe he was just out of country. So what we have is soil being what I call a vital living system. It is an organism on its own with billions of cells that are functioning. That is the key. It's a vital living system. If you destroy the nutrients that it needs to live on, you're going to lose those organisms. And then what we're finding is we're losing that nutrient use efficiency that they provide. Okay, I'm going to give you a little bit of history of the work that's coming to be presented by Soledad that I came across that really stimulated the work that we started doing when I arrived at this company. This is a production by Murphy and his group in southwestern Australia, where they grow a lot of wheat. And what Murphy did was he wanted to identify the constraints that produce in yields of wheat in Australia. And what he did was he took a bunch of fields, I think he did 40 fields, and he went to sites that were highly productive and marginally productive, or he called it underproductive. And he measured a whole bunch of factors that are associated with this ecosystem. I won't get into. But the key is he measured climate, he measured agronomic management, soil physical, chemical, biological properties. And the key here, and this is really important, everything was related to grain yield. Okay, and this is what we came up with too. So we kind of copied this, but went a couple of steps further than Murphy did. But what they found was that when they did all the statistical correlations, the most significant benefits between high and low producing sites, by the way, the differences in the production size was 1.7 versus 3.2 tons per acre. So the underperforming sites were producing half as much, even though they were a very short distance apart. But the most significant correlations came with microbial biomass and potential mineralizable nitrogen. Now that comes in many forms, and I won't get into it, but that could be in the form of microbes that are making nitrogen, or it could be organic materials that release nitrogen. But here's the take-home product. So of the 40 paddocks, and remember that word if you ever go to Australia, it's not a field, it's a paddock. That we have nitrogen, fertilizer, and clay content explained only 9% of the variability in double the yield. 9%, not very much. Potential mineralizable nitrogen explained 21% of the differences between the two sites, and microbial biomass explained 30%. So when you put these two together, you find that 51% of the differences in yield between a highly productive wheat field and a poorly productive wheat field was 50% based on biology. We thought this was really interesting, and Soledad will tell you how we transferred this to corn. Okay, so this is, I don't know if any of you have seen this book by Lynch. It's called The Rhizosphere, and Lynch explains that there are a whole bunch of beneficial things going from here to here that are associated with the microbiome. You know, things like symbiosis, antibiosis, biological control, and top among them, nitrogen fixation, growth promotion, soil stabilization, and water uptake. These are all beneficial. 7 are good things. I spent most of my life studying these 2 things, infection and phytotoxicity, because that has the biggest impact. But if you look at the amount of money that we pour into diseases, that's where the 2 factors fall in, and almost all the research. And then there's a bunch of variable things I won't get into, but this is kind of interesting. And I'll talk to you a little bit about where these things have been identified as supporting a huge level of productivity in agriculture. Okay. The first concept that biology enhances plant growth came in the 1850s. And you've got to read the really interesting things farmers did to produce nitrogen-fixing rhizobia in their crops. It was, they actually took soil from fields that had no rhizobia, dug it out, and took it to fields that had rhizobia. So they moved huge amounts of soil. Well, we all know that these nitrogen fixers are absolutely essential to a very large group of plants called legumes. You know, this includes peanuts and beans and so on. And a lot of trees are legumes also. So, but this is not an accidental thing. There are at least 26 genes and an unknown number, and many unknown numbers, that are associated with how this bacteria gets to the plant, infects the plant, produces the nitrogen in the plant, and there's the same number of genes probably in the plant that recognizes this bacteria and allows it to come and invade and set up shop in its ecosystem. Okay, so here's the good news. Very early on, breeders recognized this interaction because there's a phenotype. The phenotype are nodules. So breeders knew that if a bean plant didn't have nodules, get rid of it because it's essential. Now imagine if these same kind of interactions are occurring in corn and wheat and all the other crops that we grow. There's no phenotype. So how do we know that breeders have not completely eliminated all these genes that are required for microbial functions in our crops? We do not know that. And there may be a complete need to go backwards, and I'll show you that's happening. Okay, so here's rhizobia. This is infected by a beneficial rhizobia, one that's highly functional. And here's one that's infected by one that is incompetent. It's not producing nitrogen. You can't tell these two organisms apart. They're genetically identical. But one is not doing what it should, but yet 95% of the nodules that are formed in many legumes are these guys. And why? Well, it's a, so here's the benefits of symbiosis and yield to 21 Bradyrhizobium japonicum strains. Okay, so what you see here is 21 strains, and this is the yield. So here is the control, and you can see this is inoculate. This is the control. This is the yield you get without rhizobium, about 1,400 kilograms per hectare. And this is the best USDA strain called the USDA 110, and you can see that it pushes the yield up to about 2,700 kilograms. Just adding rhizobium to your bean plants can double yield. Cost $5 an acre. Can you get better return on your investment than that? Okay? But here's many of the rhizobia that you can see that they inoculate, but none of them produce as much yield. But look at these 5 groups. They do nothing. They inoculate, they produce nodules, they make functional things, but they don't return anything. So what you have in the soil is a competition among strains of rhizobia among themselves. There's a population of rhizobia just like in this room, and we have good guys that are highly functional, and then we guys that are incompetent, and we guys that are cheaters. So what is inevitable is that putting microbes into soil and onto plants doesn't always give you the response you want because they have a lot of competition from native organisms. So it's not a simple thing as I'm going to put microbes onto my seed and hope that they emerge and inoculate my plants, because the minute they come out, there's all these other guys that are competing to get into the plant. Now, when I was in school, they told us rhizobia infect only legumes, only and then along comes results like this from Egypt. They found that rhizobia colonized rice plants, right from the bottom to the very top. And in fact, they found that they're strain specific. Rice cultivar A may be inoculated by rhizobium A, but not by B, C, and D. So we now have not only competition, but we have strain specificity. But look at this. they inoculated very large areas of rice plants with they grew 5 varieties of rice, 7 strains of rhizobia. They did this over 5 years. And these are at sites that are ranked as the highest rice production areas in the world. So these aren't those trivial sites where you can go there and just add anything you want and you'll get a yield increase. And their average yield increase was 19.5%. 47% was the highest. Now we did the microbiome of potatoes. I did a 10-year study with Australia, and the top 10 organisms we found on the roots of potatoes was rhizobia. And we're finding the same thing on corn and so on. So it will turn out that we have a lot more work to do with rhizobia for use in other crops than just legumes. And then we thought that the nodules were just infected only with the rhizobia, and then we're finding, no, it's produced by a whole bunch of other organisms also colonized inside those nodules. In this case, it's an organism called Klebsiella that's been identified to be inside those nodules. So it's not a sterile nodule with one population. It's got a bunch of other organisms, and they're interacting, and we don't know what those things are doing either. Okay, so I'm going to tell you the biggest story of all, which is Brazil, because it's been mentioned, Dean mentioned Brazil. Brazil has been growing sugarcane since the 1590s. They grow 14 million hectares. And it's their #1 source for energy. 40 million hectares. It's not trivial, that's for sure. Back in 1970, Brazil decided to become independent of imported oil, and they decided to make this crop their #1 crop. And this lady, Baldani, was selected as the leader of the group, and she did something that is rather remarkable in agriculture. She went to the poorest farmers in Brazil, and she selected strains that were grown without nitrogen for 250 years and were still highly productive. And from this, she was able to selection was based on no input. How many of our cultivars are selected with 0 inputs? How many are selected with maximum inputs? All of them. So this may be another thing that we're going to have to go backwards. Okay, so the early research found that the microbes inside the plants were what allowed them to produce. Okay, so they were the first ones to identify these endophytes. The biggest one is Gluconocetabacter diazotropicus. There's a company now out of England that is bringing it to market. It will be in the United States available this year. I think they registered it in most states. It's called Exotic Technologies. But there's a whole bunch of other ones, including, these are all nitrogen fixing. Azospirillum brasilense was one of the first keynote organisms. But all of these bacteria have been shown to be present in coffee, pineapple, sweet potato. They can solubilize zinc, phosphorus, produce oxidants, antagonize pathogens. All those things I showed you in the original slide of the 7 beneficial functions. All right, nitrogen use efficiency. Here's the amount of nitrogen fertilizer used per hectare in various countries. In the USA, Hawaii, and Florida, where they grow sugarcane, 300 to 400 kilograms of nitrogen. In Brazil, 50 kilograms. Now multiply this by 40 million hectares and calculate the load on the environment and the cost to growers, how much you save. And this is where true nitrogen use efficiency really kicks in. So selection of cultivars is going to become really important in the future. And the beautiful thing about this is when you calculate the energy return, the dollar invested and the dollar return to the grower, the average in Brazil is $9.2. So if you spend $1, you get $9.2 back, and optimally you get 11.2. And that's terrific. I think in North America we're about 2 to 1. Okay, I'm going to talk a little bit about disease suppression, because that's, so there's 2 types of soils that we know of. Soils where disease occurs, and soils where the pathogen is present but disease does not occur. And these soils are referred to as suppressive soils. And nobody could quite understand what the heck was causing these suppressive soils, where you have a lot of pathogen but no disease. Until along comes this man. His name is James Cook. He published what I call the 3 cookbooks on biological control. And what you're seeing here, this is the Pacific Northwest wheat production area. And this is a zone that's being fumigated with a fumigant called methyl bromide. It's basically sterilizes the soil. And then right beside it is the soil that's not fumigated, and it is being impacted by a disease called take-all. I don't know if you guys have take-all here. If you grow wheat, I'm sure you do. And then he's standing in the field 7 years later after continuous wheat production. He came along and said, we must grow wheat year after year after year. And believe it or not, that made wheat production sites disease suppressive. Continuous wheat production, which goes against all logic that was available. And what they discovered, the functionality, was by a group of organisms called fluorescent pseudomonads. These are bacteria that secrete a whole bunch of antibiotics, and these pseudomonads are known to be beneficial globally now. And you can actually measure your soil health by measuring the genes that are associated with how many genes you have for the production of these antibiotics in your soil. And we have probes for those kind of genetic functions. And if you have a lot, you're going to have a disease-suppressive soil. And if you don't, you're going to be finding you have a lot of disease. So there's many ways of inducing disease-suppressive. One of the best ways is compost. Many of the composts tested are highly disease-suppressive, and adding them to soils can give you 100% disease. This is in a greenhouse substrate, and you can see that adding additional compost suppresses Pythium take-all disease. Adding it to turf, like here, this is with an organic amendment, and here's a dollar spot with the untreated, and then this sprayed with 10 pounds of compost per 1,000 square feet. And from what the managers of golf courses claim, that this saves them about $600 a year by just applying it. Plus, you don't have to put up the big pesticide platforms that are on the golf courses. Okay, I'm going to talk a little bit about slow-release fertilizers. So these are organic amendments that are used. This is a product I developed with one of my cohorts. It's made from poultry feathers and blood meal and some other meat and bone meal and a bunch of other things that we put. And it was sold by one of our big retailers. It lasted for a couple of years. We called it once-a-year fertilizer because one of the things about organic fertilizers is they are slow release. It takes them a long time to release their nutrients. And one of the other things about them is they significantly increase biomass. So you can imagine that these organisms in soil are starved, they're hungry, and you put in a carbon source of nutrients, they will go crazy and they will start eating them and their population goes up. Now it's calculated that at 10 to the 7th bacteria per acre foot of soil weighs about 30 pounds of nitrogen. If you increase that to 10 to the 8th, you now have 300 pounds of nitrogen. If you increase it to 10 to the 9th, you have 3,000 pounds of nitrogen reserves in your soil. And this is why organic materials really enhance soil, because they build up the microbes. And these microbes are available to you very much the same way as your money in the bank. You put it away, and if you got $3 billion in the bank, you're going to be okay, but if you only have 30 bucks, you're not going to even eat lunch. So one of the projects I had was working with the swine producers of Ontario because they wanted to find some beneficial use of swine manure. And one of the things we found that applying, so this is going to fields that had very high incidence of potato scab. I don't know if you guys ever seen potato scab, but it's a nasty disease that makes potatoes almost unmarketable. And we tested a whole slew of chemicals to control this disease. Here's the control, and then here's the 3 products that work best. And one of them was fish emulsion, and the other one was liquid swine manure. And not only did they increase or decrease the disease, but they increased the yield by almost double, both of those products. And we thought, well, the organic nutrients in these products are going to be the mechanism of how they do this. good food source for microbes. And then we found no. It was actually a large product due to chemicals that are present in both products. And these are what are called volatile fatty acids. And so what they are is formic acid, right here, acetic acid, you know that is vinegar, and propionic butyric acid. And you can see that fish emulsion has very high levels of both acetic acid and formic acid. So does liquid swine manure, 285. In fact, putting 5,000 gallons of swine manure per acre is equivalent to putting $2,000 worth of vinegar onto your field. That's what we calculated. So, one of the mechanisms that this works by is the acetic acid, as you know, is a biocide. It actually kills organisms. Here's verticillium resting spores plated on plates that had no swine manure, 1%, 2%, and 4%. 4% swine manure completely killed nematodes, verticillium, scab organisms, but only in soils that were acidic. Because when you put it into a basic soil, those acids are neutralized and have no functionality whatsoever. But in acidic soils, they're terrific. So there's two problems with this technology. One is there's only a limited source of swine manure. The farmers that are near them have acquired them all and they're using them. And the second is we don't have that many acidic soils, nor do you want them very acidic to use this product. Okay, so I talked a little bit about how microorganisms feed plants, and we thought that microorganisms are really reservoirs, like in a bank. As they die off, they release their nutrients back to the plant. And then along comes this paper just published, what, 2 months ago? What these guys discovered is that the root tip microbes are actually ingested by the plant. They're taken up actively, and then the plant releases a whack of enzymes on top of them, called superoxides. And then these microbes travel up the root. So what these superoxides do is they strip all the nutrients from the bacteria. And the plants, then they exit through the root hairs. So here's the microbes exiting through the root hairs. And this is actually eating your bacteria. Plants eat the bacteria. And it's called the rhizophagy cycle. And this is kind of new to us. It was rather remarkable that roots have the ability to ingest bacteria and eat them and then throw them out once they've been digested partially. They're not completely killed, but they're stripped of all the iron and zinc and so on and other nutrients that the plant requires. And there's some pictures of this going on. I won't get into it just to save some time. Okay, so on roots, we have a whole bunch of interactions. Now, the microbes interact with plants, but they interact with each other more than with plants. And these interactions is a whole bunch of communication going on. They're talking to each other. I don't like you, I don't want you near me, get away, or no, come, let's live together. I'll give you some of my stuff, you give me some of your stuff. Now, believe it or not, a lot of these interactions are by what are called volatiles. These are gases. I was trying to isolate some of these compounds for 20 years and was unsuccessful because it's a gas, and you have to actually trap this gas. And now, so the guys who discovered this, what you have down here is an agar plate, and this plate has, right about here, a plastic barrier that separates the two sections completely from each other. And what you have are bacteria that are applied on one side, and this little plant indicator called Arabidopsis is grown on the other. And what you see here is two of these bacteria. This is a Bacillus amyloliquefacciens. I don't know if you guys have, I think you used some of that. And then there's a Bacillus subtilis. And you can see that the plants on this side are very much greener, darker, bigger than the plants on this side where there's no bacteria or a fake bacteria that doesn't do anything. And here's the growth effect compared to the control. It's almost a tripling. So this volatile gas acts as a hormone to plant growth. it also acts as a nutrient source. Here's a bacteria. This bacteria, Pseudomonas fluorescens. I told you they produce a lot of chemicals. This one produces a compound called dimethyl sulfide. Now this is actually used, I think it's present in garlic in large concentration, and it's been developed into a kind of fumigant. But what you see here is plants grown in the absence of sulfur, and then plants grown in the same zero sulfur, but in the presence of the bacteria. And you can see that these plants are suffering from sulfur deficiency here. They're quite fine there. But these where the bacteria then is separated by this barrier have no sulfur deficiency. So we're just starting to discover these kind of things, and how to manage these production of these hormones will be a very key element. But some of those organic materials that you're putting in, they actually stimulate and upregulate these kind of phenomena. They stimulate stress, and this is going to be an important component of what I'll finish with. And what you have here is bacteria with bacillus. This is in the presence of high amounts of salt, and you can see that these plants, in the salt, 100 millimolars of sodium chloride, so table salt, and you can see that they're very much struggling. There's nothing on this side. And here's a bacteria, a Bacillus subtilis, growing, and then look, complete remission of stress. Pretty amazing, and it's by gas. It's by gas. We're not quite sure what all the gases are, but this is one of our best growth promoting strains in the world. This is a potato tissue culture. No bacteria, look at the root biomass that's generated. Again, this was the organism I was trying to find the active ingredient. It turns out to be a gas. Okay, so these volatile organic compounds that are now becoming well recognized, and we're starting to understand, can relieve salinity, drought, iron deficiency, and sulfur starvation. And we do it at very low levels. And these are functions that are present in these microbes that are going to be very important for crop protection. Okay, so we're at a conference, and somebody says that if you grow corn and soybeans, it's good, but if you grow corn, soybeans, and wheat, you're guaranteed to get a 10 bushel increase minimum. Why would wheat give a 10 bushel increase? And then we came across work done out of Japan. This is sorghum, and just in case you saw those pictures of roots of plants that were pulled out of the ground, well, that's not all the roots that are there. Look at the roots that you actually produce. Pretty amazing, huh? And what sorghum does, it produces two compounds that inhibit bacteria that convert ammonium to nitrate. Now, imagine soil is a very negatively charged environment. Ammonium is very positively charged, so it's held by soil really tightly. Nitrate has two negative charges, and it's repelled by soil. And one rain event, you lose half your product down to tons. This particular crop inhibits the conversion of ammonia to nitrate and keeps all that nutrient in your soils. Here's what wheat does. These are 50 or 60 varieties of wheat, and they were tested for the ability to produce these nitrification inhibitors. And look, some of them are highly potent. So, do we test for these kind of things as a rotation crop for? This would be a very cheap way of introducing nitrification. By the way, this effect of nitrification inhibitors lasts up to 3 years. One crop will give you a 3-year benefit. And there's a whole bunch of grasses that are used as rotation crops in some countries as a way of keeping nitrogen. So what's interesting is even peanuts can do it. It's really neat. Again, this is a technology that we need to look into further and adopt as a way of managing nutrient use efficiency, because it will keep your nutrients in the soil. Okay, the last thing I want to talk about is what is partially involved in a lot of the work that's being done at a number of companies, including yours, is the concept of biostimulants. And so biostimulants are now a $2 billion market. and they come in the form of microbials and their extracts, animal-based byproducts and hydrolysates, humic and fulvic acids, inorganic and synthetic products, and algal plant extracts. All of these are considered biostimulants. Now, in order to understand how these work, you have to imagine a plant that has a genetic potential to produce X amount of yield. Under nature, here it is. This is the maximum yield level of a particular plant. Does it ever reach that maximum potential? Never. The best we can do is about 80%. That's pretty good. If you can get 80% of the yield potential out of your product, you're doing great. Now, imagine if you have a highly stressed environment. Then you're going to get down to about 50 or 60% potential for yield. This is where your bioproducts are going to have their maximum impact. This is where what they do is a lot of these types of materials relieve the stress factors associated with plants. And I think Dean mentioned that under zero stress, you're only going to see very small benefit. But under very high stress, you may get a 50% yield increase. But under low stress, it's going to be a relatively minor effect. And one of these things they do is they enhance root hair production, root production. We see a lot of interactions of some of these materials, humic acids particularly, are very beneficial in the interactions between plants, and they impact plant growth. Also, lots of root hair production, and these can increase the amount of area a plant covers by a major way. Some of the work we're doing in this area is kind of interesting. We have contracts with companies that are looking at remediating polluted sites. They claim that in London, Ontario, a small city, relatively speaking, we have $5 billion worth of real estate that's polluted and can't be built on. And so there's a lot of money going into this area. So what you have is here, this is a contaminated soil, and what we have here is grass growing on it, and then we have one isolate that's been inoculated, and then here you see the grasses without the pollution and with the bacteria. But look at here, contaminated soil with this bacterial strain. Look at the differences in growth. But you can see that even in the control and the treated, huge benefits. In this particular isolate, again, huge benefits, but even on untreated soil, and so on. So these kind of beneficial effects are very important under sites where the plants are being heavily stressed by things that we don't even know about, some kind of contamination that we're not sure about. This paper was released this week. I won't get into it, but they call it the New Green Revolution based on using intercropping. And what they claim is that they analyzed over 1,000 publications, and they did a meta-analysis of what intercropping plants of two different types of plants together, and they found that it increased yield by about 33% on average. So they have used a whole bunch of intercropping. I didn't get to read the paper very thoroughly, but it sounds like this may be another component of the New Green Revolution that will come along. All right, finalizing this talk, we're going to come to an area where we have a new trinity of what is going to be happening in agriculture. We're going to have a plant. We're going to have the soil geochemistry, which very much impacts the microbes that are present in the soil, because pH alone can change the microbiome in a huge way. And then we have the soil microbes, and they're all interdependent. Plants, chemistry, microbes. But the most important component regulating all this is water. Water is going to be the essential element, as it is in most things. So summing up, this New Green Revolution combines biotechnology, improved microbial functions, smarter agricultural practices, and equipment as a means to bring about a better crop yields in the face of declining land that's available for farming. And so that's it. Thank you very much. And the next Green Revolution will come from underground.